Good morning, everyone. Thank you for joining me today as I start off our symposium. I would like to give you all a brief overview on my work this summer in the characterization of ribosomal protein gene families in the context of cold tolerance in the plant Arabidopsis thaliana. So why should we study cold tolerance? Well, you may know the plant maize, also known as corn, that is an important agricultural staple grown all around the world. Each year, a percentage of these crops die due to early cold exposure, which directly impacts the food supply. To study the genes that may impact cold tolerance in maize, we use the model system Arabidopsis, which is a common model organism used among plant biologists. There are quite a few advantages to using Arabidopsis, including its small size. Lots of seeds are produced during its life cycle. It has a very short life cycle, so you can do quite a few experiments throughout the year. And it also has a widely annotated genome, and scientists have been studying it for decades. So we have a lot of prior knowledge that can help us expand upon what we already know. Ultimately, what we learn from these experiments will be translated back into maize to aid farmers among changing climates and early cold exposure. So when we take a look at the maize growth season, it really depends on the region. However, if you plant too early in the season, the seedling may die due to early cold exposure. Ideally, you plant it during the optimal growth season and corn is produced at a high yield. However, if you plant the crop too late, the pollen will die and will lead to infertility in the crop, which limits yield and is not very great for the farmers that are trying to produce for our food supply. Our goal is to expand the optimal growth season for corn by giving seedlings a better cold response, allowing them to survive unexpected freezing or chilling. And ideally, we would like to allow farmers to plant their crops earlier in the season to avoid the heat stress entirely. The experiments that we conducted this summer defines chilling stress as temperatures from 10 degrees Celsius to 1 degree Celsius, and freezing from 0 degrees Celsius to -10. Another great fact about Arabidopsis is that it is very hardy in terms of cold resistance. It can be exposed to up to -10 degrees Celsius, which is very cold, and still make a recovery and go on to produce seeds. Now, upon exposure to chilling or freezing stress, plants undergo this process that is known as cold acclimation. And two very important genes to this process are COR genes and CBF genes, which I may refer to as COR and CBF genes. COR genes are cold responsive genes, and C repeat binding factor genes, also known as CBF, regulate these COR genes. You may wonder why we are looking at ribosomal proteins in response to cold tolerance, because ribosomes are small protein complexes that are used to translate proteins from RNA. Now, in preliminary analysis, wild-type maize was exposed to cold temperatures for 1 to 2 days, and then genes that were differentially expressed were identified for future study. This summer, I worked on the characterization of the ribosomal protein gene families UL16 and UL18, which are found in the large ribosomal subunit and are labeled with a capital L. Those found in the small ribosomal subunit are with an S, so this is an easy way to identify which part of the subunit they are in. Now, each of these families have a few different paralogs, and if you don't know what paralogs are, they are similar genes found within a family that have a similar code and code for proteins that have similar structure and function, but have some slight variability due to mutations in evolution. Here we have a diagram of the large subunit of a ribosome, in plants specifically. As you can see, the UL18 families are highlighted, or the UL18 protein is highlighted in magenta. And when they were analyzed in maize, this gene had phosphorylation of the proteins under cold. In orange, we have increased polysome enrichment under cold for the UL16 family. In blue, there's reduced polysome enrichment under cold. However, we did not look at the family involved in this summer. Now, ribosomes are playing a role in what is theorized as the paralog shift hypothesis. This is theorized to occur in all plants, but we are using Arabidopsis as our model for this system. To begin the paralog shift, some sort of stress must occur to this plant. This can include biotic stress, heat, but in this case, we will be looking at cold. On the cellular level, ribosomal biogenesis is occurring all the time. So this is the new formation of ribosomes within the cell. In this hypothesis, plants will swap out proteins with their paralogous proteins if they are better suited for surviving the stressor. In this example, UL16Z is being shifted into the place that UL16Y was, because that paralog will deal with the cold better. To test this hypothesis, I worked on a series of characterization experiments this summer. To start, I looked at the expression of COR and CBF genes in knockout mutants of these paralogs in Arabidopsis. And a knockout mutant is a plant that is grown without without a functional copy of the gene, so that we are able to see how the plant survives without the function. We grew seedlings of these plants and mainly did experiments on seedlings because we are hoping to apply these characteristics to small maize seedlings in the field. Now, we placed knockout plants of these genes into a 6 degrees Celsius chilling chamber. And we did this for 24 hours to measure the gene expression level. At 0 hours, so before we really exposed the plants to the cold, we took out half of the samples so that we had a baseline for each gene expression level, so we wouldn't compare a gene that was already exposed to the cold to one that had been in a chamber for 24 hours. From these samples, we put them in liquid nitrogen, which I was very excited to use for the first time, and extracted RNA from these samples. From this RNA, we synthesized something known as cDNA, or complementary DNA. Now, cDNA is formed from the template. So RNA is a single-stranded nucleic acid. And we use that to create essentially a carbon copy of a DNA molecule, which is much more stable. And we can use it for amplification purposes in things like PCR. Now, we use what is known as quantitative PCR to show the gene expression levels, because this is a specific type of experiment designed to show gene expression and how it differentiates from the normal wild type. Ultimately, we ran an ANOVA test and then did a Tukey test on the data to determine significance where P was less than 0.05 of the results. Unfortunately, for the UL16 gene family, we did not see any significant difference from the wild type in this experiment. However, you may notice this bar graph right here that seems to be really big compared to the rest of the data. However, we cannot confidently say that this was a significant result because we had a limited replicate size due to seed limitations. And one of our expression levels was around 4,000 and the other was 19,000, both of which were way more than we expected. And so we can't confidently say that this is a natural result of this experiment without replicating it first. For the UL18 gene family, the UL18Z paralog right here showed a significantly higher expression rate than the wild type at that time. Since these are knockout mutants, this means that plants without this copy of the gene may have a higher chance of survival without this paralog and have a more robust cold response. Another method of characterization that we used this summer was rosette area. And for those of you that don't know, the rosette is the formation of leaves at the base of the Arabidopsis plant that develops at the beginning of the life cycle. And since they are pretty symmetrical, we are able to use these to determine the growth of the plant and how they can differentiate from each other. Unfortunately, we were dealing with an aphid infestation in our growth chambers. And so we were using Marathon, which is a common pesticide, in the water that we treated the plants with to hopefully avoid getting our plants infested with the aphids. This, we believe, created an interesting phenotype in our germinated plants. Arabidopsis is a dicot, meaning that it has 2 leaves upon germination that are known as cotyledons. Now, since we are measuring the rosette area, and you can see one that wasn't affected right here, we were missing essentially half of the plant that we were supposed to study. So we couldn't confidently use these plants to make assumptions about these paralogs and how they affect rosette area, because half of our rosette was missing. So we restarted by planting around 20 replicates of each paralog onto plates and sealed them with micropore tape, because that is a lot more difficult for aphids to get into. So there are around 300 plates total, and each week I took pictures of these plates and used ImageJ to analyze the area of each individual plant. Now, certain paralogs that we planted were from segregated lines, meaning that we had to genotype these plants using DNA that was extracted before doing an ANOVA test on the data. From that ANOVA test, we were able to create these box and whisker plots that show that the paralog UL16Z, shown here, here, and here throughout the 3 weeks, had significantly smaller rosette area growth than the wild type and the other paralogs that were planted. So this indicates that plants without this copy of the gene will grow smaller regardless of exposure to cold, since we also saw it in the initial results. Now, to look back on what we did this summer, UL18Z knockouts have significantly higher CBF1 induction than the wild type, and UL16Z knockouts have significantly smaller rosette area growth. These experiments will be repeated for confirmation with a larger sample size, and hopefully we will not have as much limited seed availability. Maize will be grown with these knockouts of the genes to confirm our observations in Arabidopsis and how they translate into maize. And ultimately, our goal is to help farmers plant maize with higher cold tolerance, increasing the optimal growth season and preventing loss of crop. Additionally, in the lab, I aided Hua Lab member Hong Jun in transferring her transgenic plants for her experiment. I learned how to do DAB staining, which is observing the accumulation of reactive oxygen species, such as hydrogen peroxide, and leaves of Arabidopsis plants that were exposed to cold stress. I learned the basics of R programming and how to use ImageJ for data analysis. And I participated in the weekly Hua Lab meeting, where we went over journals and talked about current research projects in the lab. I would like to thank the members of the Hua Lab for their support and friendship, and recognize the grants that were able to support this project. Thank you. Do I have time for questions?
We have time for some questions. Go ahead.
Chloe? You talked about importance of cold water. Why did you choose this? So maize is another crop that has a widely annotated genome that is commonly used in plant research. We chose this plant just because it was what was in the preliminary analysis, so we can start with maize, work in Arabidopsis, and translate it back into maize. However, that does not mean that this research is limited into maize and Arabidopsis. The paralog shift hypothesis is supposed to happen in all plants, so theoretically it can be applied to any crop. Oscar?
After you transferred new plants to plates after the gestation, were you able to see any difference in the rosette area between the different paralogs, or did you not get far enough?
So we actually didn't transplant the plants. We restarted with new seeds, and we were really only able to see a difference in one paralog. And this is pretty common. You don't always get a significant result from all the things you test. There may only be one special paralog that has a slightly different result, but that is still great for science. We still need negative results to confirm our positive ones. Felix?
First experiments, how you needed more replicates. So how many plants would you have more plants would you have to plant in order to get a conclusive?
So since we had such limited seed availability, we really only used one individual per sample. So that is because we had really low germination rates and low seed availability. Ideally, we should have 3 individuals per sample, so we would almost have to triple our initial seed planting. Hopefully we can be able to do that soon.
One more question.
Zainab?
I was kind of wondering, so you talked a lot about maize and kind of the importance with that specifically. I was wondering why you use Arabidopsis then to.
So we used Arabidopsis instead of maize in our experiment because it is a smaller plant that takes up less resources, and therefore it is more cost effective to use Arabidopsis. We can use smaller growth chambers. We won't have to worry about maintaining such a large plant. And then we can also increase the scale of plants that we use. As you could see in a plate, we were able to have around 30 plants, while maize in that same area in a pot would only have around 1 plant. So we are able to have more replicates in our experiments, and it is a lot more cost effective for the university. Thank you.